Um, Hello, welcome you. So, today I'll be discussing the question which is related from the bioenergetics portion. So, this question which I have taken here is from GATE 2018 Life Sciences paper from biochemistry section. This is question number 8 from biochemistry, and the question states as the movement of protons through the F0 F1 ATPase during mitochondrial respiration is required for. Okay, so let's start with the basics. So what happens that we know that for energy production in our cells we have certain pathways that starts with glycolysis that takes place in the cytosol. So there ATP and NADPH are produced, and then further we have the TCA cycle, and there also we get some NADPH and FADH2. So those are the components which further enter. So NADPH and FADH2, as they did not convert into ATP directly, they enter into the mitochondrial matrix, and there from the movement or from the electron chain transport system, there is a movement of protons, and then there is the energy generation steps that happens. So uh, as it is mentioned, I'm mentioning that via the electron transport chain we have the movement of electrons. Okay, so if this is my uh, matrix, this is my matrix, and this is intermembrane space. This one is intermembrane space. So what happens that electron chain transport system takes place when, so we have four complexes. Three of them are in the mitochondrial membrane. Okay, so via the electron transport chain etc, the movement of proton happens. Okay, so the movement of proton takes place in this direction. Okay, and then ultimate result of that, what happens that as the proton moves from matrix to the intermembrane space, so the pH of the um, inner matrix becomes higher, and yeah, sorry, so the pH difference takes place. So the pH of the inner matrix become lesser. Okay, so there is generally one pH difference, and here, I'm sorry, this will becomes 8 and this becomes 7. So inner side most negative charge create, and outer side more positive charge gets created. So this is the charge distribution that happens, and this leads to generation of a proton motive force. Okay, because what happens that uh, that after the pumping of proton, what happened? This action generates an electrochemical gradient for protons with a lower pH value, um, which is outside the inner mitochondrial membrane than inside. So the protons on the outside have have a thermodynamic tendency to flow back into the matrix as to equalize the pH. So when the protons do flow back into the matrix, the energy is dissipated and some of it being used for the synthesis of ATP. Okay, so it goes back uh, uh, to the inner matrix via this F0 F1 ATP synthase. Okay, so the use of proton motive force for ATP synthesis is catalyzed by this ATP synthase. So this is a multi-protein ATP synthase, or we can also call it as F0 F1 complex. Okay, which catalyzes the ATP synthesis as the proton flow back through the inner membrane down the electrochemical proton gradient. So it consists of two components, okay, the F naught component, okay, the F naught component, and this F1 component, which is F naught ATPase. So the F naught component is embedded in the inner mitochondrial membrane, and this contains one A subunit, two B subunits, and 9 to 12 C subunits. And the C subunit consists of two alpha helices that span the membrane. Okay, so uh, so there is an antibiotic named oligomycin, which completely blocks the ATP synthesis by blocking the flow of protons through F naught of ATPase uh, of for F naught of the ATP synthase. Okay, so and this antibiotics blocks the F naught subunit and thus inhibit the protons movement and thus inhibit the ATP synthesis. Okay. So F naught ATPase, it is also made of 3 alpha, 3 beta, gamma, delta, and epsilon subunit, and is tightly uh, bound to F naught and protrude into the matrix. And it contains 3 beta subunits that are the sites of the ATP synthesis. Okay, and these are the sites of ATP synthesis. Okay, so at the center of this F naught ATPase is the gamma subunit. Okay, so the rotational motion of this ATP synthesize ATP by harnessing the proton motive force. So ATP synthase can also function in reverse to hydrolyze the ATP and pump proton across the inner mitochondrial membrane, and thus acts as a reversible coupling device, interconverting electrochemical proton gradient and chemical bond energies. Okay, so there is a theory for the ATP synthesis which is called as binding chain mechanism. Okay, so binding chain mechanism. So there, what happens that uh, it postulates that the ATP synthesis is coupled with a conformational change in the ATP synthase generated by rotation of the gamma subunit. The proton translocation through F naught powers the rotation of the gamma subunit of the F naught F1 ATPase. Okay, so which leads to the changes in the conformation of the nucleotide binding sites in the F1 beta subunit, and by means of this binding change mechanism the F0 F1 complex harnesses the proton motive force to power the ATP synthesis. So what happens that yeah, so then it goes counterclockwise 120-degree rotation. Okay, and then Here what happens, the three alpha beta dimers have three different states. So in one, the open state O, so here O is empty, and then the loose state L contains the ADP plus inorganic phosphate, and then the tight state T contains the ATP in the, okay. So rotation of this gamma subunit within this alpha beta uh, trimer hexamer converts the L state to a T state. The T state to an O and O to an L, and then the L state can accept a new charge of the substrate. So the T state can form ATP, and ATP, and then in the second stage, the ATP has fallen out of the O state, and the new ADP and inorganic phosphate have bound to the L state, and the ATP has been synthesized in this T state. Okay. So the synthesis of ATP. Okay. So the phosphorylation bond, phosphoenhydride bond of ATP is synthesized only in the T state. Okay. And the ATP is released only in the O state. So the free energy released on proton translocation is harnessed to interconvert the three states, and change in the conformational occur due to the rotation of the gamma subunit. Okay, so there is 120-degree rotation of gamma subunit in a counterclockwise direction, which changes one conformation state to another, and the rotation of gamma subunit relative to fixed alpha 3 and beta 3 complex, okay, occur in a discrete 120-degree step, and conformation of each beta subunit change in this order only. Okay. So that leads to the synthesis of the ATP. So come back to our question. So that it has been asked that the movement of protons through F0 F1 ATPase during mitochondrial respiration is required for. Okay. So why the movement of proton is required? So first is the increase in pH of mitochondrial matrix. That happens, but that does not, it is not related to the F0 F1 ATPase. Then changing the conformation of F0 F1 ATPase to expel the ATP. This is the correct option. Third option, we have the importing inorganic phosphate. No. Decreasing the affinity of ADP to F0 F1. No, that is also no. It is only for changing the conformation of this F0 F1 ATPase complex to expel out the ATP, because via the rotation, the ATP which is synthesized in the T state, it rotates 120-degree, and this ATP is now come to the O states, and which releases or expel the ATP with the help of proton motive force. So the B will be the correct option. To understand more, this is the NPTEL reference where you can study the glycolysis and the later stages of ETC and the ATP synthesis in a detailed manner. Thank you.